Hi, everyone. Welcome to Foresight's Biotech and Health Extension Strategy Group, sponsored by 100 Plus Capital. Thank you so much, Clarice, for joining on such incredibly short notice. We had a fallout of a speaker, and you just immediately jumped in. We were in contact a little while ago because Aaron was incredibly excited about your research. So really, really grateful for you to step in so promptly. And so, yeah, it should be a really fun discussion. Thank you also for for actually answering the questions already upfront, which is always difficult to get a speaker to do. And so to bringing people up to speed, we're usually asking folks about how their technologies interface with other technologies and what a challenge is that they want other people to solve to make progress in their fields. So we will hopefully get to that in the discussion. But for now, I don't want to take any time further away from you presenting. And so today, you will be discussing from nanotech to living sensors, unraveling the spin physics of biosensing at the nanoscale. And I'm really happy about this because this is one of the types of topics that is definitely bridging two of our groups. This one is the Biotech and Health Extension group. We also have the Molecular Machines and Nanotechnology group. And so I think what makes our community quite interesting is that there's much happening at the intersections of these technologies. So thanks a lot for joining, for bridging our worlds, and super excited for what you'll present to us, and then excited for the discussion. So you take it away. I'll let more people in. I'll be in the chat, and I'll have more to say in the chat. But for now, you go.
Awesome. Hello, everyone. And you can probably see my screen, right? Please
Yes.
shout if not. Okay. Please feel free to hop in with questions. Feel free to just unmute yourselves and ask quick questions if you will. But I will dive right in. I like to call myself a quantum engineer. This means that I build apparatuses to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So people get sort of confused when I say that I study things that might be happening inside birds and butterflies. So before I explain where I'm going, I really need to tell you where I come from. So even though I'm going to start talking about hardcore quantum mechanics, by the end of the half hour, I will have talked about things of biological relevance, such as organismal migration, how our cells respond to oxidative stress, to radiation. And I hope I might have started convincing you that I actually think we can learn with nature how to build better technologies. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at the nanoscale. Usually I like to motivate this whole talk about sensors because I think that humankind likes to measure things better because this gives the impression at least that we understand them better, and we might actually do. So we might, for example, try to measure better frequencies. This is a atomic clock at NIST that defines very well what a second or what a hertz, a frequency hertz, like one single hertz is based on atomic transitions. You can think about measuring better magnetic fields so that the image of your baby is better resolved. And yes, this is a magnetic resonance image of a baby inside the mother's belly. And you can think about, say, measuring better acceleration so that your gaming experience is increased, is improved. Such tiny accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity that you want to measure is very, very small? I'm going to argue that in this case, you need a tiny little sensor that can measure tiny little things. If the sensor is tiny, let's make it very tiny. Let's make it quantum. And actually, there's a whole body of mathematical proofs that can be used to show that if you use a quantum object, such as an electron or a molecule, a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. I'm going to describe a little bit a technological quantum sensor that I work with in the past. And the reason why I'm talking about this technological quantum sensor is because I'm going to be making comparisons with a living quantum sensor further down the line. But this is the promise that there's a quantum sensor, magnetic quantum sensor in diamond. Okay, In fact, it's a electronic spin in diamond. So let me talk about spin for a bit. So spin is a purely quantum mechanical property of objects that is as fundamental as charge and mass. Electrons have spin, atomic nuclei have spin, and spins measure how well the quantum object, say an electron, interacts with magnetic fields. Usually chemists and physicists and engineers depict the spin with an arrow. So spin up or spin down, two different states of this spin property represent two different energies of interaction with magnetic fields. So the promise is I will put a single electronic spin in diamond to work as a magnetic field sensor. And here's how this works, right? So at a particular crystal effect in diamond at room temperature that I won't have time to describe, a crystal effect provides a relatively isolated electron in the diamond lattice, and this electron has a spin, okay? This effective spin, which actually lives in this lattice effect, is very special because it has quantum state dependence fluorescence intensity. Now this is jargon. This is jargon for the people of technological quantum sensors. This effect, it can be excited by light. It can, when you shine a laser at this point in space, it absorbs the light, gets excited, and then fluoresces, emits lights back. And it turns out that this crystal effect, just by looking at how strongly it's emitting light, how high a number our fluorescent counts it is in the lab, we can actually determine if the spin is up or down. This is a very special property. It doesn't happen very often, but I need you to know that this exists in technological quantum sensors. So you have this material, diamond, and just by exciting it with light and by counting how many photons you are receiving back, you can actually read a quantum property, which is this spin state of that particular electron. But let me show you how this works as a very sensitive magnetometer, right? So here to the right, I plot what I call 0 and 1, which in vertical axis is energy. So those two states just represent the different energy levels of spin up and spin down. And in that electron, the difference of energy between spin up and spin down is in the microwave range, about 3 gigahertz. So it turns out that the state that is marked 0 is insensitive to the presence of magnetic fields, whereas the state marked 1 is sensitive to the presence of magnetic fields. So if the diamond is immersed into a magnetic field, state 0 will not care, whereas state 1 will actually be promoted by a tiny quantity called delta. Okay, and it turns out that this delta, via a physical interaction called the Zeeman interaction, delta is proportional to the magnitude of the magnetic field that the diamond is immersed in. So that, and now this is a very important point that doesn't often get made. The problem of measuring a magnetic field is actually mapped onto the problem of being able to measure a little mismatch delta from a known energy difference, right? And actually, this is how all technological quantum sensing, if you're curious, work. There is a certain quantity that you want to measure, magnetic field, electric field, temperature, and you know that a certain energy difference shifts in a known way as a function of that property. So you being able to measure that property is mapped onto the problem of you as an experimentalist being able to measure this mismatch. And actually, this is a signal processing problem, and engineers and physicists have known how to measure mismatch from resonances from many, many decades. So that this is how that spin in diamond works as a technological quantum magnetometer. Okay, without giving a lot of details, here's the big picture, right? The big picture is to have a tiny little sensor inside a nanodiamond that you can bring in very close proximity to a tiny little sensor a tiny little sample, and be able to measure tiny little magnetic fields produced by this sample. Okay, so this is why people care about those technological quantum sensors. Okay, so this is what was just a necessary introduction. Okay, because this technological quantum sensor is not what I really want to talk about, but I actually will be using it as an analogy later on. It turns out that up to this, up to like 5 years ago in my career, those were the types of sensors I was used to dealing with. But at some point, I realized that nature engineered sensors that many times and very often outperformed humankind-made sensors in crazy ways. I, in particular, am going to talk about a magnetic sensor that is naturally made, and that I'm going to argue is quantum enhanced, and that works pretty similarly to the defect in diamond. And I changed slightly the title of my talk, but this is what I really want to talk about. I'm faculty at UCLA in electrical engineering since 2019, and as I mentioned, I build instruments for a living, and if you build instruments, December of 19 was not a good time to start your group because the pandemic hit. We're super delayed, but we're getting there. And I am very happy to share our vision and to start conversations with people in the biosciences. So, but to start talking about naturally made sensors and biology, right, I need to talk about biology at the nanoscale, namely the chemistry. It's actually been known from many decades in a field of chemistry called radical chemistry that magnetic fields can alter the final products of a class of chemical reactions that actually are sensitive to electrons spin. Okay, so here's how this works with my hands, right? There's a certain class of chemical reactions that happen, and at some point the chemical reaction comes to a crossroads. Okay, and then the chemical reaction waits to see or to sense the electronic spin of a certain electron in a protein, say. And it turns out that if this, and now I'm going to use jargon, I don't expect you to understand that, but for those of you who might have some background in physics, maybe you understand. If that particular electron that we care about has its spin measured by the environment to be up, the chemical reaction continues through one branch. If the certain spin of interest has its spin measured by the environment to be down, the chemical reaction continues through another totally different branch. The macroscopic final products of those two branches are different, okay? And magnetic fields can actually change the probability that the electron spin is up or that the electron spin is down. So a very fast interaction with the magnetic field at the crossroads of the chemical reaction while it's waiting to see the spin state of the spin can actually have macroscopic consequences, right? Because the products of the branches are different and at much longer time scales, because this is like a downstream process. And this phenomenon, again, has been demonstrated in test tubes at room temperature, in solution in the gas phase, in the solid phase state, and for magnetic fields as small as, say, the magnetic field of the Earth. So here's where the biology comes in. And to talk about that, I will need to start talking about birds. I really don't want to talk about birds, but I need to start talking about birds because historically that's how it all started. And that's very cool to think about it, actually. So since the 60s, birds have been known while they migrate to follow the magnetic field of the Earth, at least as a partial cue. Nobody doubts that. There have been like many behavioral experiments that say, yes, the birds are using the magnetic field of the Earth to migrate. It's sort of crazy if you think about it, because the Earth's magnetic field is smaller than the magnetic field that you sense when you put your cell phone close to your face. So it's really tiny, right? So how might they be doing this? And there have been many different theories and all. And at some point in the end of the 70s, a brave cohort of theoretical biophysicists proposed something that at that point was a pure heresy. Okay, say they hypothesized, and at that point was a mere crazy hypothesis. They said, well, were this same type of spin-sensitive chemical reactions be happening under physiological conditions in the bird somewhere, right? Maybe birds and organisms in general might be sensing magnetic field to the extent that they might be sensing different physiological concentrations of products coming from these spin-dependent chemical reactions. At that point, that was just an idea, right? And it turns out that experimental biophysicists started looking for proteins that could sustain such spin-dependent chemical reactions. And so before I talk about the particular protein that people found, for birds, they had an idea that maybe, one way that the bird could be sensing the different physiological concentrations of products from that particular chemical reaction is, for example, if that chemical reaction that is independent is sort of related to the photosensitivity of the bird's eye. For example, if the bird's photosensitivity could vary as the bird head moves left and right, the bird could actually be able to see magnetic field lines just because of this modulation in photosensitivity. While this particular hypothesis remains to be established or disproven, experimental biophysicists started to look for animal proteins, because at that point this was relevant to birds, animal proteins that could sustain those spin-dependent chemical reactions. And at that point, the only animal protein that could sustain such spin-sensitive chemical reactions was a flavoprotein that is called a cryptochrome. So cryptochrome is present in the retinas of birds, in the antennas of migrating butterflies, our retina, and it's actually present in all our cells because it also has circadian rhythm regulation functions. And actually, cryptochrome is very well conserved throughout the tree of life. All those organisms there are known to express cryptochrome, and many of those organisms there have been shown macroscopically to have some sort of magnetic field sensing ability related to this cryptochrome. Okay, the ones in box in red are those for which there have been some studies relating magnetic field sensitivity and cryptochromes. And yes, humans are right there. And it turns out that evidence for this type of cryptochrome-based magnetosensing, experimental evidence, is now really, really widespread, but unfortunately at very disconnected length scales. Okay, let me talk first of all about the macroscopic evidence. Okay, so for example, at very large length scales, there are behavioral experiments with birds, where people, like during migration season, take like 20 birds and put them into cages, change the magnetic field around and move and try to track where the bird wants to go out. Flies do not migrate, but I don't know how people can do this. This is harder than quantum mechanics for me. Flies can be trained to find food in the presence of a magnetic field. So the flies are trained to do this. But then the researchers remove the cryptochrome gene from the flies, and the flies are no longer able to find food based on the presence of a magnetic field. In a further experiment, the researchers put back human cryptochrome inside flies, and the flies are back to being able to find food when there's a magnetic field. So those are just two examples. There are many, many more of experiments that are consistent with a spin-dependent chemical reaction happening in vivo. However, the next level of evidence, like at slightly smaller length scales, this evidence is all the way through to chemical length scales, which you see to the left of my slides. Like for cryptochromes in solution in a test tube, there is no doubt that those molecules are working, are sensing and interacting with magnetic fields in a way that is very related to the way that the electron spinning diamond was uh, uh, measuring and interacting with magnetic fields. So, for example, I mentioned that for the spin in the electron spinning diamond, just by looking at how strongly that lattice effect was emitting light to us, was fluorescing upon laser excitation, we could actually infer if the spin was up or down. For cryptochrome, it's actually the same, just by measuring. So cryptochrome has a pigment, which is a flavin. Cryptochrome is a flavoprotein, and a pigment, a chromophore, is usually fluorescent, right? So when we shine laser at it, it gets excited, and then it gives us light back. It fluoresces. And for cryptochrome, depending on the spin, if the spin took the chemical reaction took one branch, the fluorescence level is 1. If the spin took if the chemical reaction took the spin down branch, the fluorescence level is another 1. And this is exemplified in the picture to the top left there, where you see the fluorescence emitted by cryptochromes in a solution as a function of the time during which we excite the tube of cryptochrome with the laser. That is, we want to promote fluorescence. The first thing that you see is that the fluorescence is decaying. This means that you're bleaching your sample, you're killing the pigments, the laser is strong. But as the researchers pulse a tiny magnetic field on and off during this experiment, the fluorescence reacts following the magnetic field, as you can see in the inset. This is not an experimental bug. This has been tested many times. So what we see here is that depending on the state of the magnetic field on and off, you can see different intensity levels, which is proof that a tiny magnetic field can actually alter how many, on average, how many spins take this pathway or this pathway, which is exactly how a quantum sensor works, a technological quantum sensor works. And actually, there's many other similarities with technological quantum sensors that I'm not going to discuss. But there's a problem here, right? It's very hard to say, well, the bird is doing its thing because there's a quantum-dependent chemical reaction happening inside it, right? So people don't talk. Chemists don't really talk to experimental behavioral biologists, and nobody can prove anything unambiguously. So that's what we're starting to do in our group. We want to bridge those two length scales. So to cut a long story short, what we want is a bottom-up approach. We build something that I would describe as glorified microscopes with coils. Coils to provide the magnetic fields that can actually change the pathways of chemical reactions, in a microscope so that we can start looking at such phenomena, say inside one cell, then 2 cells, and at some point maybe a tissue or maybe an embryo, right? To see if we have a better handle at how spin physics might actually be doing things, influencing things macroscopically, the downstream. Something that's just for the experts. What we want here in our experiments is to correlate 2 different regimes of fluorescence. The first regime of fluorescence is for very weak fluorescence coming from the autofluorescence of those chromophores that can sustain spin-dependent chemical reactions. And that fluorescence happens very fast, from nanoseconds to microseconds. So we want to be able to prepare the spins in a certain way, measure that we're preparing the spins in a certain way, right? We prepare the spins using the coils, and then correlate what happens using regular fluorescent markers that take place, that are much stronger in fluorescence and that happen at much longer time scales, from millisecond to second. And those will be the first experiments at the nanoscale that actually correlate spin physics with downstream things in a living cell. Okay, so that's what's unique. So basically, what we're doing is actually hijacking instrumentation from the field of technological quantum sensing and applying the same technology onto those electron spins in proteins by treating them as bona fide quantum sensors, because that's what they are. They are bona fide quantum sensors. So, and when I say that the instrument that I'm building glorified microscopes, the microscopes that we're building, they won't look like this. They really look like what you would encounter in a quantum sensing, technological quantum sensing experiment, in an atomic physics experiment. It's a big optical table with mirrors, electronics around, and it's just that it's working on a biological sample. This is not our experiment. We are behind. We're not there yet, but that's exactly what this will look like. And actually, this modality of quantum biology, spin flavor of quantum biology, is really limited right now by the lack of the type of high-tech instrumentation that we want to deal with. Okay. Again, the experiments in this field that exist right now are either very chemical or very biological. There's nothing wrong with those. Those are hard, beautiful. And we also think that instrumentation will push the envelope by trying to see those phenomena, those quantum phenomena in vivo. And very importantly, we want to be able to control such phenomena in vivo. If there's any physicists in the audience, what I think how I would describe to you what we're trying to do is coherent control of light-matter interactions when matter is in fact biological matter. But this is not a talk about birds. I'm almost finished. This is absolutely not a talk about birds. We needed to start talking about birds because historically that's where it all started. But the same type of spin-sensitive chemical reactions are now being shown to actually underlie many, many types of metabolic regulation phenomena. For example, the production of reactive oxygen species depends on magnetic fields in a way that is consistent with it being spin-dependent. Cellular acidification rates, cellular respiration, cellular glycolysis rates, as seen for a plate of cells under a metabolic analyzer, are also magnetic field sensitive in a way that is consistent with the spin model. Stem cell mediated growth in planaria, yield of DNA repair, the autofluorescence from mitochondrial flavoproteins seem all to be magnetic field dependent in a way that is consistent with a spin model. And I want to spend a couple of minutes to describe this last picture there, because I think you're going to get a kick out of this story, right? So I have a friend who's a vanilla precision measurement physicist. So what he does for a living is build very good Faraday cages. There is chambers that can shield external magnetic fields very, very well. Okay, so the magnetic field of the Earth is about 50 microtesla. So inside one of his chambers, the noise level is about 1 nanotesla, so orders of magnitude smaller. So he was hearing those things about, well, magnetic fields in biology. So here's what he tried. Again, it's a physics group trying to do bioexperiments, but these experiments have been replicated by people in Mainz in Germany and Tufts in the US. So they grew for 2 days tadpoles inside those hypermagnetic chambers. 2 days, because that's as long as they could go without asking for like a biolicense of some sort. And under one condition, they applied inside the hypermagnetic field, the hypermagnetic chamber, they applied a tiny magnetic field that mimicked the magnetic field of the Earth. And in the other condition, they did not apply any internal extra magnetic field, just the nanotesla level. What they saw macroscopically was that the tadpoles grown under the mimicked magnetic field of the Earth were macroscopically okay, whereas the tadpoles grow under just the nanotesla level, 30%, and that's what you see in the picture, are macroscopically deformed. Okay, so this is sort of crazy because you didn't need to add a magnetic field, and actually you just subtracted a magnetic field, right? And the magnitudes of the magnetic fields involved are so tiny that everyone's best guess right now, there is no other mechanism that seems to be able to explain this unless it's related to some sort of magnetic sensitive chemical spin dependent magnetic field dependent chemical reaction. And this, I think, opens all sorts of Pandora boxes, right? So what's the magnetic field in Mars? So can we grow lettuce in Mars? Can we reproduce in Mars? Or how can we use like those endogenous quantum mechanical degrees of freedom to actually drive physiological outcomes? And for reasons that I won't have time to explain, but reasons that chemists and physicists understand, these spin-dependent chemical reactions only happen at relatively small magnetic fields. Okay, so magnetic field from like magnetic field of the Earth, like microtesla to say 10 millitesla, which is relatively small. This means that if you put the cryptochrome vial inside a big magnet for magnetic resonance imager, it won't behave as a quantum sensor anymore. Okay, so those things only kick in at small magnetic fields, which can be good because we don't need huge magnetic fields to engineer ways to change physiological outcomes. But it also means that we really need to understand what those freaking things are doing to us, right? Because all our electronics equipment, they are emitting some sort of low intensity electromagnetic radiation, and we really need to understand what might be going on. So this is what we're starting to do to explore in a very quantum inspired way in our group, and that's very new. We want to go beyond that bottleneck. We really want to start doing quantum-like experiments in biology to either establish or refute or establish down to a certain noise level whether quantum mechanics can really account for physiologically relevant biological phenomena, right? And be manipulated to say therapeutic advantage. Again, can we engineer magnetic fields if we have an index? Oh, and another thing that I didn't mention, right? So even though if just started with cryptochrome, now it's known that many other molecules, again, reactive oxygen species, other proteins, usually with a chromophore, other proteins also can sustain the same type of spin-dependent chemical reaction. So if we have an index, oh, for this protein that controls this reaction, this is the magnetic field that we need to apply to tweak it the way that we want, can we start, it's not going to happen in 5 years, in 10 years, but in 50 years, can we start using tiny devices to steer physiological outcomes using those endogenous quantum mechanical degrees of freedom, right? This would be some sort of like quantum optogenetics, right? No marking needed. The degrees of freedom are there. We just need to know how to control them. And also like to technological advantage, right? So if nature really is using quantum mechanical degrees of freedom to function, it knows darn well how to deal with noise. And the reason why we don't see quantum mechanics in our daily life is because everything that is big is pushed back to classicality. And that's also the reason why we don't have very stable quantum computers, for example, yet, because they start quantum, but at some point, uncontrolled interactions with the environment starts kicking in, and they are pushed back to classicality. I won't explain too much about that, but I'm not saying like let's replace Google's beautiful quantum computer with a molecule, but are there strategies that nature is using to overcome noise, quantumness quenching noise, that could be applied on top of existing quantum computer, quantum sensing architectures, for example? So I think there's a lot in this field, a lot of interest from people who are thinking about senescence, stem cells, embryology, immunology. So, and I think that we have a very different approach to all of this. I would like to finish by thanking my group members who started in the group during a pandemic in a new lab in a theme that is still very misunderstood, right? And I always like to finish by saying, may the quantum be with you. What are your questions? And let's.
Wonderful. Thank you so much. This was a whirlwind. Thanks a lot. Thanks a lot for joining us. You were definitely squaring many of our groups here. Before we have lots of audience questions already. Before we get into them, I would love to know from you a little bit. We asked you via email, as we do with all of our keynotes, the questions: what challenge would you like to see solved for progress in your field? We're currently building our tech trees and our ideas to map different challenges that different people want to see solved to actually make progress in your field. So you already answered this a little bit via text, but would you be keen to answer this also a little bit in person before we jump into specific questions?
Sure. Again, I'm very partial to this answer, right? Because that's what I do for a living. But I truly believe that the current bottleneck in this, in quantum biology in general, is the lack of quantum people in the field, is the lack of people who actually build instruments, right? I think the next frontier is start treating biological quantum knobs as bona fide quantum objects, quantum sensors, right? And in order to do this, we need to hijack instrumentation from the field of quantum sensing, quantum computing, right? So this is the challenge that I think needs to be solved now, so that in 50 years, we might have a Star Trek-like little thing that we apply, put a magnetic field and very predictably drive those spin-dependent chemical reactions.
So what is like a very specific challenge? Is it just more people in the fields or more interactions between the fields? Is it like specifically people working on very specific tooling? Like, do you have like something?
Oh, so you're getting to my pet peeve. So first of all, we are in the middle of nowhere, which is cool. I really think that interdisciplinarity is going to solve. Only interdisciplinarity is going to be to solve big questions in science. But we are in between physicists, biologists, and chemists. Biologists have been tweaking physiological outcomes chemically for many decades, right? That's like the whole pharmaceutical industry. You give chemicals and you induce chemical reactions. So when you talk about using electromagnetic stimuli, it's a little bit weird. Biologists are not familiar with that, and one of the things that I need to do is educate biologists that this is a thing, and help show them that maybe those endogenous quantum knobs can be tuned. The other problem are the physicists. So physicists, those are the worst, because sometimes they don't even want to engage with me, right? Those chemical reactions that are spin dependent, they are known to happen at like under physiological conditions, like more or less at room temperature and stuff. And the physicists, they just say, well, we're not familiar with this, it's never going to answer. They shut down. And that's very sad. So this is problem number one, right? And that's one thing that I am trying to remediate in my quantum biology center at UCLA, because we have an arm that's thinking about curriculum development, community building, on like how to train the next generation of people who actually can think about quantum biology, right? What do we need to do now to have a quantum biology department in universities like in 20 years? The other thing that I will rant about is the fact that there's no funding, right? Two program officers at NIH already told me more or less in these words, like one actually used these very words: NIH is not ready for quantum biology. And then you're like, okay. The other thing is the fact that it's a no aging field, and that's actually very serious. Most people pivot to doing quantum biology experiments in the second half and the last third of their career when they're established. They have some sort of secure funding stream, right? So there's zero incentive for young blood to enter the field. Like the groups that exist, we're sort of relatively well funded already, but the groups that survive, they're not super well funded, so it's very hard to attract talent, to attract and keep talent. So I mean, part of my being here talking to you is to help change like all those aspects and to wear my hat of community builder in quantum bio.
Okay, wonderful. I think I would love to have Aria's question in there next, and then we'll go to Larry, who raised his hand, and to John. Aria, do you have a question that's super relevant to what you just said? So I think.
Yeah, it's a bit of a loaded question. Hi, Clarice. We know each other quite well, actually. But yeah, I did want to bring up that topic that most of the capital for this field is coming from nonprofits, foundations, private money, and even that is fairly limited. There's no federal financing for this, which is, 90+% of the research done at research institutions is funded by the federal government. So yeah, I think maybe, I don't know if Clarice wants to dig into that a little more with the wider group as to how this group could maybe work on that problem or think about other funding sources. I'd love to hear her take.
Yeah. I think what's needed is some sort of concerted effort, say, for one flavor of quantum biology. I'm partial to spin quantum biology because I think that if you're familiar with other flavors of quantum biology, I think that spins in biology live longer than other quantum phenomena. So I think that if nature is using quantum to function, doing this via spins is a very good bet. I think there needs to be some sort of concerted organization that has as a goal, let's give 10 very well-funded years to a select group of investigators that complement each other. And during those 10 years, let's try to push this so that within 10 years, we open the door for, we will have results for there to be, and a proof of concept things, so that the government will feel better to dig in, young blood will feel better to dig in, right? So that there needs to be some sort of, and that's why I think talking to all of you is important, right? There needs to be some sort of coming together of funding of a select group of scientists that can turn the table for once, be well funded enough to pursue those things in a way that, so just like I spend 30 hours, I work way more than 40 hours a week, but I spend about 30 hours a week just writing grants, which in the end is like a waste of my time. And I think, and actually not, I think I have a about half of the grant getting rate than like the usual in the nation for like NSF. So odds are already stacked against me. And like NSF, if you apply for an interdisciplinary thing at NSF, sometimes they make you like go through two different panels. So it's the law of like multiplication of small numbers, right? You have to please both panels. So there needs to be a big something that changes this for a short time so that we can establish the field and help build momentum so that it itself perpetuates a little bit more after that.
Wonderful. That was very eloquently said. At Foresight, we just got a bit of funding. I mean, it definitely wouldn't do the entire trick, but to do more experimental physics and quantum research and to fund a few fellows in that and to serve a little bit more like a cover academic early stage organization that is somewhere in between academia and more startup world and to provide researchers with a bit more cover without taking as big of a chunk of a cut of their budget as I think other universities also do. So anyway, I would love to also follow up on that. But at the same time, that is obviously nowhere near what I think would be required to even make any remote progress on this. And so thanks a lot for stepping out there and joining us to talk about this. Okay, next one up, we have Larry, and then I'll go for questions in the chat.
Yeah. Hi, Clarice. I was wondering, how does this technology differ from squid technology that, a few years ago had a lot of, kind of hype or hope for, imaging and things like that? Do you see this being an imaging technique where you wouldn't need superconducting, or I mean, the claims they're picking up magnetic fields, probably due to electrical activity, but but how would you foresee this differing from that you have
Okay.
different
So
cycles and squid, or yeah.
two parts to your answer. So I'm going to talk a little bit about the technological quantum sensor that I talked about in the beginning of my talk, that electronic spin in diamond. The advantage of that as compared to squid is the fact that it works at room temperature, right? That it for example, what people can do, it's different from what we want to do. But what people can do is shoot tiny nanodiamonds inside cells, and those nanodiamonds contain a single one of those particular electron spins. And using the quantum sensing protocol, they can get information, say, about temperature inside the cell in a quantum enhanced way. Doing this is good to probe biology, but doesn't say anything about biology being quantum or classical. Okay, but it works at room temperature. So a squid might be more sensitive because it works at low temperatures. You can do materials characterization, but it's not good for biology. So this is how that technological quantum sensor can compare with squids. What we're talking about here is less, one could use those, say, spins in cryptochrome as very sensitive magnetic field sensors, right? By putting cryptochrome and trying to measure its fluorescence, for example. But if you want to do this, if you want to use the spin degrees of freedom of cryptochrome as a magnetic field sensor, use the nanodiamond, because nanodiamonds are easier to work with. The point here is rather using those endogenous degrees of freedoms in cryptochrome and in other molecules to do something to the physiological reactions that they're attached to. It's a fundamental new way of thinking about like metabolism and physiology, right? This is the power of this whole thing. And that's different from this technological quantum sensing business. This is using those things that work as natural quantum sensors and use their sensing. I mean, if they can sense a magnetic field, they can be controlled by a magnetic field, right? Use those endogenous properties towards a certain specified physiological outcome, right? And this is very different. This is really like quantum optogenetics, right? It's using electromagnetic radiation of a particular frequency, intensity, direction to macroscopically alter things that are connected to those spin-sensitive chemical reactions.
Larry, any comments?
No, I mean, it's just, I mean, what, I mean, you're not talking about delivering a lot of energy to flip things and things like that. I mean, it's just, I'm not exactly clear that to me seems a bit of ways off, but, perhaps possible. But, I mean, there are a lot of free radical reactions and
Exactly.
metabolic pathways, and, a lot of flavon things. And, I mean, but
exactly. And there's a lot of evidence. None of the evidence is conclusive. It's either in solution or like with plates of cells, with whole birds, that DNA, yield of DNA repair, how much of your DNA gets repaired, that the production of reactive oxygen species, all of those things are controlled by weak magnetic field, stem cell growth in planaria. All those things, they are controlled by magnetic fields in a way that is consistent with an underlying spin quantum model. So those things exist. Those things have been seen. They have just not been seen at the same time, like the spin physics and like the macroscopic signature of what's going on.
Wonderful. I have Steve Fox next, and then we'll go down the chat.
My basic question is about whether biomagnetism research has been extended beyond ferromagnetism into areas like paramagnetism or diamagnetism, that kind of thing. And whether or not spin stable elements and isotopes have been studied in terms of their mechanism of action in, let's say, protein centers or enzymes, to identify that as a potential quantum bio connection.
Okay. Again, many things that I need to say. First of all, let me talk about enzymes. Enzymes seem to function via another quantum phenomena. Okay, there's a quantum phenomena called tunneling, and the tunneling of protons and electrons through enzymatic sites seem to be what makes them work. This is work by Judith Klingman from Berkeley, from chemistry from the last 30 years. So enzymes only work because of this quantum stuff. Okay. The other thing is that there are some isotope effects that have to do with spin in more or less the same way that I described, but I don't know enough about them, and I don't think about them enough. But there are people actually thinking about using those for like quantum pharmacology and things like this. The other part of your question had to do with permanent magnets. Okay. Organisms are known to be sensitive to magnetic fields in three different ways. One is the way that sharks do it. Sharks and rays, they have like conducting tubes. And the idea is that as they swim, they cross magnetic field lines, and they crossing the magnetic field lines induce a voltage in those tubes, and they can sense the magnetic field via differences in Lorentz force. That's one way. Bacteria, magnetostatic bacteria, can feel magnetic fields because they have big chunks of lines of magnetite crystals, permanent magnet crystals, right? They're very, very chunky. And the idea is that those crystals, they just align with the magnetic field. And people think that this alignment might be messing up with mechanosensitive ion channels. It's not super clear, right? So for permanent magnets, like magnetite crystals, for little magnets to be used by organisms as a magnetic field sensor, they need to be chunky, okay? Because if it's a tiny sensor, a tiny magnet, the thermal energy, right? Everything is sort of wobbling around because of thermal energies. If the crystal is small, the tumbling will be will erase the effects of the crystal being actually aligning with the field. Okay, so for complex organisms such as butterflies, birds, and turtles, what people think is that if birds were using magnetic fields in a way that a magnetostatic bacteria does, people would have found those big chunks of magnetite, and people don't find those big chunks of magnetite. The other thing is that people have made experiments where they put like a big magnet on the bird's beak, I think, and the birds do not lose the orientation. That is, they're not following this magnetic field that is close to them. So whereas bacteria seem to be using permanent or paramagnetic centers, that does not seem to be happening inside birds and cells, given the present evidence. Yeah.
Lovely. Next one up, we have Abdulkar Ramo, and perhaps you want to say 1 second about yourself, because I think you're new to the group. Welcome.
Yes, nice to meet you. I am actually a biochemist by training. I'm not a physicist, but I always was interested on trying to understand the limits of knowledge, and try to be comfortable at that area. So I'm from SMS Biotech. I'm the Chief Scientific Officer. And I would like to ask you about possible equivalence of cryptochromes in bacteria. And the second question is, I would like your stance on quantum entanglement, if you may tell us that. Yeah, thank you.
So one analog of like the one protein that can sustain spin-dependent corrections bacteria is photolyase, DNA repair enzymes.
Okay.
And I'm sure there are many others. That's one that I remember, and one of the papers that I flashed there actually is doing exactly that. It's using photolyase as a magnetic field sensor.
So
yes.
Yeah. So if you have internalized bacteria with possibilities for example, cryptochrome effects, do you think there will be an interaction between the eukaryotic cell and the bacterial cell? Is that something?
I don't know, and I think there's so much more that needs to be established before. Right. Right now, we need to go from the ground up, like working, like with say bacteria overexpressing photolyase and just looking at what happens, right, in a plasmid or something. There's so many things that need to be established or refuted before we can start talking about interaction. So I don't know. I don't have a best guess yet.
Now, your official stance on quantum entanglement, if you may.
So there are many quantumness levels in life, right? So there's the basic quantum zero level, which is the fact that everything is made out of atoms, which are quantum per se, right? The next level, quantum 1.0, I would call, is coherence. Coherence is a property of a single quantum system. And coherence allows the single quantum system, say a single electron spin, to be in a superposition of up and down. Okay, so this quantum 1.0 is what technological quantum sensors at their minimum use, and that's the type of things that those living quantum sensors that I described are using to sense. Okay, so I am almost convinced that nature is using some sort of quantum 1.0 there. Quantum 2.0 is entanglement, which is a very organized interaction between two or more different quantum objects, right? Some people have posited that entanglement might play a role in biology. Given the current evidence, I am not convinced about this yet.
Okay, wonderful. Thanks for leaning out of the window here. If Alex Zaronkov is still here, you can shoot out your question if you're interested.
So sure, thank you. Hi everybody. Alex Delling from New York. I'm just wondering, are there any well characterized signaling pathways for cryptochrome? Do we know what happens downstream when there is an excitation by magnetic field? And
No,
do you have any gene expression data before and after?
So it's not known. And that's exactly the bottleneck, right? A bird is too complicated. We don't know what happens downstream in a bird because, well, there's signaling and stuff, right? So this is what we want to do with our like single cell experiments, right? We think that in a single cell, we might have a better handle by using different fluorescent markers, like of what is linked to different spin states in cryptochrome and in other proteins that have the same characteristics as cryptochrome. I am super happy to talk to someone from bio who might have other ideas on how to connect those two things, how to connect spin physics in cryptochrome, which we can measure using fluorescence, with like how we can distill what happens downstream. I'm very curious on how to do this, maybe not using fluorescence for the second part.
Gene expression before and after. So we can look at transcriptional response, methylation response.
Awesome. So
for example.
before, so it's like before exposure to magnetic fields and after.
Yes. And then there are probably even data sets out there that we could use anyway, because that's how you study the effects of magnetic fields on biology in general. It's just there you need to look at the also excitation levels of cryptochrome as well. Maybe not even necessary, but yeah, it would be interesting if you have data sets.
Would love to do this. And on another vein, there is research done by the Army in 2016. It doesn't talk anything about spin, but it talks about weak magnetic fields. And they can show that before and after weak magnetic field irradiation, there is a difference in epigenetics. There's a difference in methylation levels of DNA or something. So yes, there are epigenetic things that people are thinking about. I am not familiar with them, and I would love to collaborate with someone who could help us, who could give other ideas, because we were the instrument part. We would love to understand more of the biotech part. Thank you.
Well, I think you came to the right group. It's really wonderful to have had you here. I know we're 2 minutes over time, but I don't want to leave you go without the final question that everyone gets asked, which is, what could this group do to help you tomorrow? And I think that you already, made some step at answering this, but is there like a very concrete, specific ask?
Specific ask is, if possible, lobbying for quantum biology, right? It goes from lobbying to quantum biology to providing me and my colleagues with support. I mean, it can go from like student support to instrument support, right? There are many things that I think, and expertise, as I have already, I mean, as I just discussed with Alex, right? So there's so many things, and like, I'm always open for discussion. Discussing with people from the biosciences, biotech is very useful. And I really think that there's a lot of biotech things like in 10 years, a lot of biotech startups that could revolve around the idea of using magnetic fields, using electromagnetic radiation to actually drive specific phenomena in biology, stem cells, senescence, embryology, and so forth. I just feel bad for John, because John has been trying to ask a question for us. Can we give like 10 seconds to?
Hi, can you hear me?
Yes.
Nice to meet you. I was wondering, when I saw your description of the Faraday cage with the defective development of tadpoles, whether this might have implications for the safety of space travel, both for the astronauts and for the plants they're growing, either in interplanetary space or on planets with no magnetic field like Mars. And it also occurred to me in relation to the questions about funding, whether maybe NASA or the European Space Agency might be a good source of funding.
Yes, I've been trying to talk to them and to Elon Musk. So if anyone has a line to Elon Musk, please let me know. No, seriously, people have only been thinking about microgravity, and I don't think we can separate microgravity from magnetic field effects. You're absolutely right. What happens in a multi-generational flight in space, right? What's the magnetic field? I have tried asking what's the magnetic field that people sense in the space station. I didn't get a reply. So yes, I think this is should also be of extreme interest for NASA and stuff. There's just not people thinking about it, and I'm trying.
Well,
Now,
maybe you should
The
join.
space station is inside the Earth's magnetic field, so I would
I
expect
didn't
very
know.
different results in interplanetary space or on Mars.
Right.
Well, Clarice, I think you have to join our space group as well. We have both a
Oh my
nanospace group,
God, I didn't know it. Oh
and
my God.
that
Yes.
is mostly, I mean, there's a few NASA folks in there that are, I think, quite interested in these questions. So maybe that's your next presentation. You can cycle through from one of our groups to others. There's not
Yes.
really a single one that it doesn't touch. For now, thank you so much for joining us. I will probably also be reaching out about a few follow-ups. We're doing a workshop later this year with a few more, I guess like exotic approaches for making progress in longevity in biotech. And so this would be a really great fit. So hopefully we have some follow-up to do. Okay, everyone, thank you so much for joining. This was really fun. It was really fun to see many of you in person at the biotech and Foresight meetup here last weekend, and virtually the day after. We have a few more of these coming this month, and I mean, they're monthly now, but I think Clarice just added her email in the chat for those of you who are interested in following up with Clarice. And yeah, you know where to find Foresighters. We're usually on the Discord for asynchronous communications. And so thanks a lot, Clarice, for joining. It was really like a true pleasure to have you. I think you really have blew some minds here. So thanks a lot. I hope you all have a lovely rest of your day and see you next week.